Welcome, everyone. Let's start with Staphylococcus, a group of bacteria that look like clusters of grapes under the microscope. This unique shape helps us recognize them and understand their behavior in infections. So, Staphylococcus bacteria are gram-positive cocci, meaning they stain purple and are spherical. The name actually comes from the Greek word for a bunch of grapes, which fits their clustered appearance. They're non-motile and can survive with or without oxygen, which makes them quite adaptable. Now, diving a bit deeper, Staphylococcus belongs to the Micrococcaceae family. There are many species within this group, but Staphylococcus aureus stands out as the main one causing disease in humans. Most other species usually live harmlessly on our skin. An important distinction is between coagulase positive and coagulase negative types, which tells us a lot about their potential to cause disease. Understanding Staphylococcus classification is key. Coagulase positive species like Staphylococcus aureus tend to be more pathogenic. Coagulase negative species such as Staphylococcus epidermidis and saprophyticus are often less harmful but can cause opportunistic infections. Some species aren't pathogenic at all. This classification helps us predict their behavior and guide treatment. When it comes to how these bacteria cause infections, the source can be external, like from other patients or carriers, or internal, from bacteria already living on our bodies. Transmission happens mainly through direct or indirect contact, including contaminated objects or by inhaling airborne droplets. This explains why hygiene and infection control are so crucial. Focusing on Staphylococcus aureus, it naturally lives in our nostrils and on the skin. Morphologically, these bacteria are gram-positive cocci about 0.5 to 1.5 micrometers in size. They form those characteristic grape-like clusters because they divide in 3 planes. They're non-motile, don't form spores, and some strains have a capsule which can help them evade the immune system. The cell wall of Staphylococcus plays a big role in its ability to cause disease. It has peptidoglycan, which gives rigidity and activates immune responses. Teichoic acid helps it stick to host cells and protects against immune attacks. Protein A binds antibodies to avoid being engulfed by immune cells. The capsule and clumping factor also help the bacteria resist immune defenses and clump together, which is important for infection development. Staphylococcus produces several extracellular enzymes that boost its virulence. Coagulase helps the bacteria resist immune cells by converting fibrinogen. Hyaluronidase breaks down connective tissue, aiding spread. Penicillinase breaks down certain antibiotics, making treatment harder. Catalase neutralizes harmful hydrogen peroxide. Other enzymes like DNAase and lipase also contribute to bacterial survival and tissue invasion. Toxins are another critical part of how Staphylococcus causes disease. Cytolytic toxins destroy various cells, damaging tissues. Enterotoxins cause food poisoning symptoms. Epidermolytic toxin leads to skin conditions by splitting epidermal layers. Toxic shock syndrome. Toxin triggers a severe immune response leading to toxic shock. Each toxin has a distinct role in disease manifestation. The infection process starts when bacteria enter through damaged skin or tissue. They then stick to host cells or the surrounding matrix, avoiding immune defenses. Once established, they multiply rapidly, eventually causing tissue damage. This sequence explains how Staphylococcus progresses from colonization to active infection. Staphylococcus causes a variety of skin infections. Impetigo is highly contagious and causes blisters. Boils are painful lumps filled with pus. Folliculitis inflames hair follicles. Wounds and burns can get infected, leading to complications. Styes affect eyelid glands, and pustules are small, pus-filled bumps. Recognizing these helps in diagnosis and treatment. Beyond the skin, Staphylococcus can cause deeper infections affecting various systems. This includes osteomyelitis, meningitis, endocarditis, septicemia, and urinary tract infections. Respiratory infections like tonsillitis, pharyngitis, sinusitis, and pneumonitis also occur. These deep infections often require more intensive treatment. Intoxications happen when bacterial exotoxins cause disease, either produced inside the infected host or in contaminated food. Key intoxications include food poisoning, toxic shock syndrome, and staphylococcal scalded skin syndrome. Understanding these helps us grasp how toxins contribute to disease beyond direct bacterial invasion. Staphylococcal scalded skin syndrome progresses as certain strains produce epidermolytic toxins that break down connections in the skin's outer layer. This leads to blistering and peeling, with milder forms like bullous impetigo and more severe forms like Ritter's disease in newborns. Typically, the condition resolves within about 10 days, but it can be serious. Toxic shock syndrome toxin, or TSST1, activates T cells massively, causing a flood of cytokines that trigger inflammation and systemic symptoms. It acts as a superantigen, bypassing normal immune processing. This overwhelming immune response is what leads to the severe effects seen in toxic shock syndrome. Clinically, toxic shock syndrome starts with localized bacterial growth, followed by toxin release into the bloodstream. Early symptoms like fever and nausea appear, progressing to severe systemic problems including low blood pressure. Skin peeling, especially on palms and soles, occurs, and in severe cases, multi-organ failure and shock develop. Early recognition is vital. Laboratory diagnosis involves collecting specimens like pus, swabs, blood, or urine, depending on infection site. Smears are stained simply and with gram stain. Cultures grow rapidly on common media, producing characteristic yellow colonies and showing beta hemolysis on blood agar. On McConkey agar, small pink colonies appear. These features help confirm Staphylococcus presence. Biochemical tests further identify Staphylococcus. Catalase is positive, while oxidase is negative. The bacteria ferment sugars like glucose and mannitol, producing acid but no gas. Coagulase test differentiates pathogenic species. Antibiotic sensitivity testing guides effective treatment choices, ensuring the best outcomes for patients. Treatment options include beta-lactam antibiotics like penicillins and cephalosporins, which target bacterial cell walls. Quinolones offer synthetic alternatives. Sulfonamides combined with trimethoprim are also used. Glycopeptides, such as vancomycin, are reserved for resistant strains. Choosing the right antibiotic is crucial given resistance concerns. Epidemiologically, Staphylococci are primary parasites of humans and animals. They are a frequent cause of hospital-acquired infections, often resistant to antibiotics. Post-operative wounds commonly get infected by these bacteria. Cross-infections occur through carriers in hospital settings, highlighting the importance of infection control measures. Controlling MRSA involves isolating infected patients to prevent spread. Screening hospital staff helps detect carriers who may unknowingly transmit the bacteria. Antibiotic stewardship is essential, using antibiotics carefully to reduce resistance development. Together, these strategies are key to managing MRSA in healthcare environments.